Hello, everybody, and welcome to this webinar on webinar series on biotechnology and agriculture. So today, my guest is Professor Mohamed HIJRI from University Mohammed VI Polytechnic in Morocco. And I'd like first to thank him for accepting the invitation and for also sharing with us one of his research topics. But before starting on this, I would like first to thank you for the participation to the webinar. And thank strategic partners, OCP-Africa and RUFORUM. So I would like again to renew my thanking to Professor Mohamed HIJRI. And also, I would like to introduce him. So, Professor Mohamed HIJRI received his PhD at the University of Burgundy in Dijon, in France. He is currently full professor at the University of Montreal, Canada, and his sabbatical year at Mohammed VI Polytechnic University. Professor HIJRI has published more than 100 peer-reviewed articles, including prestigious journals such as Nature. As you know, Nature is one of the most cited journal within the scientific community. In 2006, he received a Leaders Award at the Canada Foundation for Innovation. And in 2012, he has been awarded by the Natural Sciences and Engineering Research Council of Canada for the best academia-industry partnership. Professor HIJRI is an associate editor in Frontiers in Microbiology, Mycorrhiza, Microorganisms, and was an invited editor in Frontiers in Plant Sciences. His research focuses on mycorrhizal symbiosis, microbial genomics, and environmental microbiology. His lab uses omics-based toolkits, microbiology, molecular genetics, and field research to understand the role of microbes on plant growth and performance, as well as resilience and sustainability of agrosystems. Thank you again, Professor Mohamed HIJRI, for accepting my invitation. And the floor is yours from now. Thank you.
Thank you, Aziz, for the introduction. So I'm really happy today to take you into a tour in Cropland Microbiota. So first of all, I will give you a background on some definition, introduce you some concepts before giving an example of a case studies of my research. So microbiota versus microbiome. So if you consult literature, you will find both these terms that they are used commonly. So now what they mean. So microbiota normally refers to a microbial communities in a given habitat. So in soil, for instance, microbes do not live alone, they live in a community. And this is really important to understand. For microbiologists, since a long time ago, they are dealing with the community rather than individuals or population or species. So microbiota is the appropriate term to use for a microbial community that lives in habitat. In a given habitat, we know biota. However, microbiome refers to the microbial genomes. So, and as you can see in literature, these terms, they are used interchangeably. However, for French-speaking audience, microbiome may refers to the habitat. And this is really particular for French languages, but in English term, microbiome refers to genomes of microbes, or what we use, it means metagenomes, a group of different genomes that are coexisting in a community. So yesterday I have done just a basic search in PubMed using these two keywords, microbiota or microbiome, or both. As you can see, this graph shows the number of publications per year. So before, I will say 2000, we had typically like less than 10 publications per year, and starting from 2000, then we have really an exponential growth, and now we are reaching almost over 12,000 publications a year in which they used microbiota or microbiome as a keyword. But of course, 2020 we are now not reached the full citation number. So now, what explains this massive increase in terms of number of publications per year? Of course, this is directly linked to development and advancement of next generation sequencing or high throughput sequencing that I will be using in the example I will be giving you. So now, in microbiology, typically, as I said, we are not really talking about species or taxon or whatever, but we are using typically two terms. So depending, we have some other variants, but we use OTUs. That means operational taxonomic units. What that means? An OTU is an operational definition that used to classify sequences that you have obtained from conventional sequencing, but also from high throughput sequencing. Typically, we use a clustering method that means with sequence homology higher than 95%. Of course, this threshold might vary depending on the group, but this is really the standard. The sequence homology higher than 95% defines somehow what is likely as a species if we compare it with higher organisms. So recently, we are using another term, which is ASV, that means applicant sequencing variant. And this term, it's typically used in a different bioinformatics pipeline. Instead of grouping or clustering sequences with percentage of homology, we are correcting using algorithm to correct sequencing and PCR errors. And then we have this applicant sequence variant. What is the advantage of this? So this applicant sequencing variant, we don't do any clustering. That it means you can retrieve any data from the databases and then you can compare studies. However, those that they use OTUs, so it's really depend on the cutoff that have used. So we can't really compare different studies to do meta-analysis. So please refer OTUs. Sometimes you will find Z OTUs or M OTUs. So this is typically quite similar. Now, core microbiome, what means? So core microbiomes typically is defined as a minimum community of microbes that is essential to a good functioning of ecosystem. So the concept first was introduced by the gut microbiome. They are much more advanced than soil microbiome. And one of the important questions, is there any core microbiome in terms of taxonomic composition or soil core functions? Regardless who is present there, what we are interested here, it's really for the function versus the taxonomic composition. As you can see here in the right, we have different ways, different methods to determine core microbiome. So as you can first here, we have sampling, presence and absence of OTUs, and then we compare different communities. So shared ones, we can call them a core microbiome. And here we are, instead of using presence absence, we are using dead abundance. So the percentage of occurrence within the community. We can also use phylogeny, but also OTUs that are shared across different communities. So this is really an example to illustrate this kind of core microbiome. So this study has been done on 3 cultivars of Brassicanatus seeds. So these three cultivars, you can see him with different color. These names are here. And as you can see, if we overlap, if we compare these three communities or these three cultivars, it was that they share all a third of the OTUs of the microbial taxa that they are present. So then we can, if we use the definition by absence presence, then this groups that are presented here is dominant by alpha protobacteria, donc represents the core microbiome. Then we can push things further, and using exactly the same sample, we can define what is eco-microbiome. So eco-microbiome is something really special. It's a community that lives in a given habitat. Here, for example, for the avatar cultivar, so it has its own microbiome, the microbial taxa that they are only present in this cultivar and they are not present in the others. And also we can see here, as I said, the core microbiome. Together, all these ecobiomes, then it forms what we call a pan microbiome. So a pan microbiome is sometimes broad, so of course here the authors have used only 3 cultivars. So a pan microbiome is really we have to compare as much as possible different cultivars to have a broad idea on pan microbiome. So now, the developments in high throughput sequencing. Of course, as I said on the graph, the impact on the publication number is mainly due to advancement of high throughput sequencing. As you can see here, it's a really an overview of the platforms and the technology that they are available. As you can see, you have here the read length, and you have the amount of the sequence generated per run. So the conventional sequencing technology, it's called Sanger. As you can, it's still really quite used today. So in terms of read length, it's quite good. We can even reach 100 base pairs per sequence. However, we can only obtain 1 sequence per reaction. And typically, if you have an instrument, we can have a 96 well placed, then we can have 96 in one run. Compared with other, this is a gigabyte per run, and it's a logarithmic scale. If we compare with the high-seq, you will see that this is really far away for comparison. We generate a huge amount of the data that we have to handle using bioinformatics. So the most used today platforms are Illumina platforms. So the one I will be talking today is MySync, which is not present here anyway. And each platform, of course, over time, it evolves by increasing the amount of sequences, but also improving the sequence length. We have new technologies that they are really going for long reads that are mainly used to improve genome assemblage. So now, plant microbiomes. So the plant microbiomes are mainly composed by bacteria and fungi. And for those who are interested in plant disease, I would say for plants, more than almost 2/3 of the plant disease are caused by fungi. So fungi and bacteria are the most important group of microbes that interact directly or indirectly with plants. So where they live? They live partly in all different organs of the plant. We have what's called phyllosphere, so the microbe that they are living in leaves, in fruits, or in shoots in the upper ground. Then we have below ground communities, and typically these communities are much more complex than the phyllosphere microbiomes. We can find the rhizosphere microbiome. How we define a rhizosphere? Typically, it's soil that is influenced by root exudate. So it's soil surrounding somehow the roots. Then we can go to rhizoplane. It's really a very thin interface between the root surface and soil. Then we can have root or in the endophyte or endosphere. These are typically endophytic communities that are living inside the roots. We can also find endophytes in the upper part. So this is an example of a leaf microbiota, where you can have here a trichome, and we have here bacteria that are typically living, looking as forming biofilms. We have here another community of bacteria. This is typically a fungal hyphae. We have other structure that they look like fungal conidia and other microbes. So in terms for root microbiota, as I said, they are much more complex. So if we decompose them, we can have, of course, beneficial microbes, some biotic microbes, but also pathogenic microbes and microbes that they are free-living and they are interacting with plants indirectly. So this is an example of nodules that they form as a symbiotic association with legumes, roots. We have ectomycorrhiza, typically for shrubs and trees in forests. We have endomycorrhiza that are much more common. Gram-positive bacteria, gram-negative bacteria, gram-positive bacteria, and also actinobacteria. As you can see, all these microbes are interacting each other. So now, since a couple of years, we have introduced a new term, phytobiomes, which is a bit different than a macrobiome, but phytobiome is much more complex, and it groups not only plants and their microbiota, but also soil and environmental conditions. So, and typically it's defined as a really being as a complex network of interactions between plants, of course, their environment, microbial communities, animals, and soil that influence the agro-ecosystem. It's much more broader than microbiota or microbiome. So communication in a phytobiome. So communication is extremely complex. As you can see here, these are really just examples. If you have here the reference, if you are interested in having more information, but typically in the roots or in the upper part of leaf, we have different ways to communicate, either by using volatile organic compounds or different signals. And you can see that all the bacteria, fungi, insects, plants, they are interacting each other. And again, the most complex biotopes is really happening in the roots. So this example here really shows very nice demonstration of the importance of microbiota in plant health. So the authors have took groups of seeds, and those seeds are canola seeds here, and they have done different treatment using in silico analysis, but also microscopic observations. And here, when they compared low bacterial diversity, typically they took seeds, they disinfect them to reduce the microbial communities or the bacterial communities, and then the high bacterial diversity. And in each of the treatments, then they used untreated or bacterial treatments. So they have implemented, they put some artificially some bacterial strains. And interestingly, the authors reported that when the microbial diversity is getting lower, then the plants is getting vulnerable with pathogen attacks. And it's same true. Same plants, when they are treated with bacterial, so they are more tolerant to pathogens attacks. So when we have the higher bacterial richness, as you can see here, the plants are doing well. So it's really a nice demonstration that microbiota or the diversity of microbiota is directly linked to health of plants. So now, just to take a couple of minutes to tell you about a new concept, holobionts. So holo, it means really something that's holistic, it's very broad, and this picture, so one of the best examples forever I can give you, it's lichens, which is symbiotic associations between fungi and algae and cyanobacteria. So if these organisms taken together, they are assembled differently, they don't have nothing to do with the morphology of the lichens, and when they are in the symbiotic phase, they form something really just amazing, completely different. So how we define a holobiont? A holobiont is typically defined as an assemblage of the host, human body, animal's body, or plants, and their microbiota. So also this microbiota, we can find symbionts, but also free living that they are interacting closely or loosely with the host. And together with the host and the microbiota, they form what we call an ecological unit that it evolves under natural selection. So it's much more complex, to define this. And I'm using here this scheme just really to show you in a very, very simple way what is the holobiont. So if you take the host alone in aposymbiotic phase, so this host is subjected to a natural selection. And if you take the symbiont in other hand, as a free living phase, it is subjected to under natural selection. However, if we put them together in the symbiotic phase, both together, the host and the symbionts are subjected to a different ways of natural selection. It's really a form on ecological units. So if you want to learn more about holobionts, of course, I recommend you this literature in which they have the principle of holobionts, and in the same way, they're hologenomes. And I will be explaining what means hologenome. And I was looking for literature, and the first term that has been used was not the holobiont, it was hologenome that was used at the end of the 80s. But then the first report was in 92, in which they have introduced the concept of a holobiont. Then these terms remain dormant for many decades, and they come out with the development of next generation sequencing. So here, with a hologenome, what that means? A hologenome, you can imagine that is the genome of the host, but also the microbiome or the metagenome. So as it's shown here in this scheme, you have the host genome, where they have a host, human or plants or any other organisms, living organisms. Then you have the host genome, then the hologenome means the host genome plus the microbiome, and in the microbiome, the authors have used different color to illustrate somehow their impact and their evolution or co-evolution with the host plant. So there are some microbes, typically those there are some or some even some pathogens, they co-evolve in parallel somehow with their host, while others, they are not really interacting directly with the host plant. So just to keep things simple, a hologenome, it's the host genome plus the genomes of their microbes or the microbiome. Now, if we push things more complex, so we have just published last year really a new concept of holobiont chronobiology. So that was really amazing. We have discovered that some fungi, particularly arbuscular macrophage fungi, that they live entirely in soil. They are not almost in contact with the above ground. And what we have found in these fungi, they have photoreceptors. They are able to sense light. And also they have a circadian rhythm that allows them to know or to differentiate between the day and the night. And with that concept, what I have introduced as a concept of holobiont, so here we are presenting tree with their microbiota. So the whole system or the whole ecological unit is somehow using a chronobiology to synchronize all this biological rhythm. And how this biological rhythm are controlled, so they are controlled through signals, typically carbon, nitrogen, and phosphorus. Of course, the plant, what they are needing nitrogen and phosphorus, so minerals, and the microbiota, what they are requiring from the host plant, they are requiring carbon that is produced by photosynthesis and going down. So this is really an example of how this ecological community is really well organized. Not only they communicate each other, but also they have developed molecular mechanisms that they allow them to synchronize their biological rhythm. And this is really a very nice example in which is a mycorrhizal hyphae, and as you can see here, this mycorrhizal hyphae, it serves really as a microhabitat in which a myriad of bacteria, they are living on its surface. You can imagine that these fungi, that they are directly linked with the roots of the plants, they are getting carbon, and it's in turn, they are giving part of their carbon to other microbes. Now, a big question. So can microbes help to feed Africa? So, of course, my question, I'm convinced, yes, and I will give you evidences and examples how microbes can feed not only Africa, but also the world. So plants depend entirely on their microbiota, on their microbes. And that interactions, it's not something new or not man-made. It has been really dated back to the origin of plants, and microbes are much more ancient than plants. So microbes play a crucial role in all plant physiology processes, particularly in harsh conditions, where a really typical, in Sub-Saharan African, where the agro-ecosystem are facing challenges of water stress and salinity. And also, by harnessing beneficial microbe, we can help to feed not only Africa, but also the world. Soil microbes. So soil microbes, as I said, play a crucial role in the growth and the health of the plant. So these are some examples that they are really well known in the literature, some indicates nutrient cycling, nutrient acquisition, the composition of organic matter, aggregation formation, gas regulation, repelling or competing pathogens, and inductions of plant defense and phytohormone production. And microbes that do all the job, also we call them PGPR, that stands for plant growth promoting rhizobacteria. But there are also other microbes. So toward the green revolution, so a couple of years ago in 2013, so the American Society of Microbiology has organized a workshop in which they have really scientists discussed how microbes, or at least to come with an inventory or with ideas of how microbes can help to feed the world. So the report is fully available in this link, and they took here the citation. So improved understanding of plant microbe interactions has the potential to increase crop productivity by 20%. So this is enormous. This is a really good percent, while reducing the fertilizers and pesticide requirement by 20% within the 20 years, according to this report. So these estimates rest with the organism that all plants rely on microbial partners to secure nutrients, better pathogens, and to resist to environmental stress. So we can find a lot of literature in the last decades. Now, how we can really manipulate plant microbes to improve crop yield, and what I mean here by manipulating, it's not GMO. We don't do any genetic modification. Manipulation, it's really just trying to use some stimulants or some substances to influence or to change the community structure. That was why I'm talking here with manipulation, but nothing to do with GMO. And here in this scheme, which is nicely represented with all the community that we have, I have shown, sorry, where we have the phyllosphere communities, but the rhizosphere communities. And for us as a microbiologist, we see that the plants has a genetic pool extremely complex. It's available for plants. And also, these communities are able to exchange their genes via a phenomenon called horizontal gene transfer, which is very, 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 very common in prokaryote bacteria and archaea. But also, we can have some events that can even happen between prokaryote bacteria, archaea, and fungi, and or even plants. So what I want to say here, that the plant have access to a pool of genes which is extremely complex and diverse. Now, some words about plant biostimulants. So what means biostimulants? Biostimulants is a term that is quite broad. It's used to describe typically substances, but also microbes that they aim to enhance plant nutrition, enhance plant growth, crop quality traits, biotic and abiotic stresses tolerant, water holding capacity, and so on. So plant biostimulants also designate a mixture of substances and microorganisms. So this is already an example of a biostimulants category. If we decompose them to the substances and the microbial inoculants, in the substances, this is not really a restricted list. There is others, but we can find some popular ones: humic fulvic acid, some protein hydrolysate, typically amino acids, but seaweed extract I will be talking about as a way to manipulate crop microbiota, but also phytohormones, chitosan, and biofilms, so on and so forth. In terms of microbial communities, so mycorrhizal fungi, fungal endophyte for living, microbes, plant growth, promoting rhizobacteria, rhizobia, and so on. So now I will be giving you an example of case studies that we have published last year, how we can manipulate crop microbiota to enhance crop yield and secure for global food security. So this is really an example. There are so many others. And in this example, we have really used a seaweed extract that is commercialized as a biostimulant or a complement of fertilization, but it's not definitively a fertilizer. So we want to see the effects of seaweed extract on soil and crop root microbiota. So this product, this project, at least was a collaboration with Acadian Seaplants, where they have registered this trademark Stella Maris that was the first in Canada that has been registered 6 years ago. So the claim was Acadian Seaplant was commercializing that product, and that product works. So as you can see here from the vine, the seaweed extract might increase by 68% of yield, which is really enormous. So for me, when I saw this graph in the beginning, it was very hard to believe that. So, and the other, questions, so this is not a fertilizer. So it's a complement of fertilization, and I was really curious to see how that works. And the Acadian Seaplant didn't really know the mechanism by which this seaweed extract acts on plants. So if you see its composition, so it contains oligo-elements, quite interesting oligo-elements, and also from zinc. As for the first webinar was with zinc, it's not that large amount, but it still has a good amount of zinc, but also boron and other oligo elements. Of course, that seaweed contains potassium. Why? Because we used KOH to hydrolyze algae. And I was really curious with this one here with carbohydrate. In addition to these elements, so they contain mannitol, fucundans, and arginic acid, but also other flavonoids. And that really interests me. This carbohydrate with this organic matter may have an impact of microbial communities. And as you can see, it is a really very poor, hardly no phosphorus, and very, very, very low proportion of nitrogen. And this is not really commercialized as a fertilizers, but is a complement of fertilization. However, in terms of organic matter, we see that there's a very large proportion between 13 and 16%. So the hypothesis was seaweed extract application increases microbial diversity and shapes microbial structure. Typically, we're interested in two factors. It's the richness, microbial richness, which we call alpha diversity, and also the community structure, which refers to beta diversity. Typically, by applying seaweed extract, we have a direct effect on microbial communities, and in their turn, they have an impact on the plant growth. So the experimental design was as follows. So the trials was a greenhouse trials, and these greenhouse trials, there were two. So they were under organic farming. So please keep in mind, this is important. So it's all the trials were done under organic farming using two crops, tomato and pepper, and using natural soil. So this soil was not sterilized, was not multiplied. It was taken directly from soil and brought to the greenhouse to set up the experiment. So we used a split block design with 8 blocks per crop. 4 tries per block. Here again, we didn't use a pot cutter because we want to have something really very close to natural conditions, although we are still controlling many other parameters. We used 4 plants per tray, and the treatments were for planted, non-planted, because we want to see the impact of seaweeds on the microbial community without plants. But for some other treatments, fertilized and non-fertilized. So these are really randomly splitted, and as here for tomato and for experiment, and both trials were done in a different times. They were not done in parallel. So first of all, we went in a research farm in the region of Montreal, in which organic farming has been done for many decades. We collected the topsoil 15%, as you can see here, the tractor, and we put the soil in these big trays, and we transport them to the greenhouses. So these are the trays with natural soil. So the soil was a loam sandy soil, and it was selected deliberately to have a very poor soil. As you can see here, it's slightly acidic soil and very, very poor in terms of nitrogen and phosphorus. So at the end of the experiment, as you can see here, you can see on the back of the picture, students, summer students are kids, starting to harvest pepper experiment. As you can see here, this is one block. So these trays were not planted, and these two were planted, treated, and no treat, and straight away we can see really the difference. It's obvious. And here again, the difference for pepper experiment, not treated, and we treat. We didn't put any fertilizers here. We didn't put any nitrogen. And as you can see here, the plants that were treated looks much better with really a very green color. And even though we didn't put any any nitrogen. And if you see at the root system with the plants that they are treated with seaweed extract, they have a rigorous root system compared to the control. So then what we have done, so on molecular sequencing workflow, typically at the harvest time, what we have done, we took the root system. Of course, we split the root system for microscopic observation, but another part was dedicated for DNA extraction, but also we collected rhizosphere soils. And this is a really typical thing routinely doing in lab, extracting DNA. So we are using two different kits, one kit for plant material and another kit for soil. So soils is much more complex and very difficult to extract clean DNA out of it. Then went for PCR, targeting ribosomal genes, 16S for bacterial communities, and ITS for fungal communities. Then we do barcoding, typically each sample we are giving a barcode, unique barcode. We pulled all the samples together and we subject them to Illumina sequencing, and followed by bioinformatics that allows us to sort somehow samples that they were pulled previously, and out of that bioinformatics analysis we can tell who are there, so their name, their taxonomic affiliation, but also we can predict on their role. So the prediction here, it's really depend on the groups. It's not the appropriate method to use it. If we want to really to know about functions, we should have done RNA extraction targeting expressed genes. So now give you a summary here of the data. So result on crop growth, as you can see here, except for one trial on tomato here, we have a significant difference between controls and amendment. As you can see here, for paper, for both tomato, and for control and treatment, and you have here the percentage. So what Acadian was claiming is actually true. We have up to three times increases of here in fruit, in terms of fruit number, except from tomato average fresh weight, because we are using this cherry tomato. That makes sense because the tomato was all quite small. That's why we didn't have any significant difference in terms of the size of the fruit. For all the other parameters, we had a significant difference between treated plants and non-treated plants. And here again, you can see, I mean, the fold increases between control and treated plants. So this is really an overview to give you an idea about the yield. So graduate students and undergraduate students were happy because they took all those tomatoes. They were really quite delicious. So they, of course, they took them home for cooking and for testing as well. So results. In terms of these are really an overview of raw data that has been obtained from soil bacteria and the root bacteria, but the same is true for fungi. A total of 192 samples, 96 of tomato, and for paper we obtained almost 10 million reads, and over we have like 5,000 amplicate sequencing variant that resembles somehow different species that represents more than 98% of reads. For bacterial soil, we have had 96 samples, 48 of course, because in soil we don't have use this planting pickers here. We are using tomato and paper plants. But from soil, we have half of the trays were not planted. In terms of richness or bacterial diversity. So if we compare between controls and amended for tomato and for pepper, soil roots and soil, here it's just really an overview. We see that typically we have same composition but with different frequencies or different abundance. So this is not really, cannot tell us if there is any significant difference or not. It's just really visual. We have done further statistical analysis to test the significance. So the same is true for fungi, and as you can see here, there are some colors that they are highly dominant compared to the other, and also we compared control and treated plants. So in terms of alpha diversity or fungal richness, I won't have time to show you all the data, so you have the reference of the paper if you are interested in more details. Typically, for fungal soil, what we have shown that we have a significant difference when we compare the planted trays between control and planted. However, when we compared trays that were not planted, we didn't observe any significant difference for tomato nor for pepper. But we had a significant difference between control and planted, both in pepper and in tomato for soil fungi. And for root fungi, we had also a significant difference in tomato. However, we didn't observe any significant difference, although if we have, a tendency, but it was not that significant. So effect on alpha diversity on bacterial richness, almost same pattern has been observed in terms of fungal diversity. No significant differences for no planted trays, significant differences between control and planted. Same is true for pepper. However, in pepper, what we have observed here, it's a difference, significant difference between control and amended tray that were not planted. Root bacteria, significant differences for tomato, but it was not for pepper. So using PICOA analysis, then to see really here the community structure or the alpha, the beta diversity that uses the abundance, or relative abundance, of course, of each SAV, then we have really a clear difference of all the samples that they were treated compared to those that they were treated. And of course, using Permanova, it's really the appropriate way to test the significant effect of the community structure. We had a really highly significant factor of fertilization. And somehow really fertilization here, I mean by application of seaweed extract, had a highly significant effect on the community structure, and somehow it shaped the community structure associated with tomato and pepper. So we have done also redundancy analysis, and I want to really go into details, but this is really very interesting, and what can tell us this kind of analysis is really the taxa that they are positively correlated with different agronomic parameters. And what it tells us, if we are interested, for example, of crop yield or crop fruit size or biomass or whatever, then we can see which taxon are associated with that parameter, and then we can go back probably to do conventional microbiology methods to try to isolate and characterize that microbe that was highly correlated with the parameter of interest. I think with this, I will sum up with some conclusions and some perspectives and take-home message. So microbes, as you can see here, and I hope that I have convinced you that microbes play a crucial role in agroecosystems. They should be considered in management of agricultural practices. Manipulation of microbiota can help to increase crop yield, but also to sustain agricultural production by reducing, of course, chemical inputs and reducing, therefore, the environmental footprint of agriculture. Of course, even though that we have done an amazing progress and advancement on the sequencing technologies, we are still really needing to generate knowledge, basic knowledge, to understand the cropland microbiota, and most importantly, to understand their resilience against the global climate change, and also using integrated approach and for long-term experiment. And with this, I would like to thank you for your attention, and I can take any questions from the audience. Thank you very much.
Thank you, Professor Mohamed HIJRI, for this the webinar, and also for having highlighted mainly what is the microbiome and what is its role within the life of the plants, and also how it helps the plants improving productivity. So really very interesting. When I was watching, a lot of people were reacting to your webinar through the chat discussions and highlighting how important for them this subject. Thank you for having elaborated on this one. So of course, we will start this question and answers session. I know that there is a lot of questions coming there. We'll try to take a maximum of questions we still have time now. I will be starting with a question from Zina Brushayed. I think you know this lady, Mohammed. She's working with you. In your opinion, what is the most sustainable method to preserve microbiota and optimize the use of plant health and yield?
Okay, thank you. They're interesting questions. For instance, the answer is straightforward. So we're reducing agricultural practices, tillage, for instance, chemical inputs, because if we are really want to manage pest management, we are using chemicals, pesticides, fungicides, antibiotics, and these products do not discriminate between beneficial and pathogenic fungi. So you are killing everything. So really, yes, we can improve, microbial biodiversity by really reducing somehow chemical inputs.
Thank you, Mohammed. A question from Reda bin Reed. So he said, Dr. HIJRI, how could omics approaches help to define a better biostimulant for plants and their microbiota?
Well, actually, it's a very interesting tool that I'm using actually in my lab, but many other colleagues they are using, because to study microbiota, so we can't, as I said, we are really talking about community, and omics toolkits is the only approach, I would say, or the only tool that we are available that allows us to study all the community integrated with host plants, but also with animals and soil physical chemical properties.
Yes, we have a question, Mohammed, from Michel Ghanem. Despite the great advances done, we are still more in a spray and pray approach as we don't know what is there. How can we use this fantastic tool in a more targeted way?
Well, actually, we know quite a lot. I mean, with these technologies, we can sequence the complete metagenomes and we can know exactly from there, and even we can discover new species, new microbes that they have never been isolated or sequenced. So this has been done throughout the time, and of course, these tools allow us really to do a real breakthrough in terms of understanding soil biology and crop microbiota. But of course, we need to do more investigation to understand more, mainly for long-term experience.
Yeah. Anyway, Michelle, it doesn't hurt to pray anyway. So thanks for the question, Michelle. So a question from Amarjit Basra. Which will be more important in the future, microbial inoculants or genetic association with microbes? Very interesting question.
Well, very interesting questions indeed. Well, personally, I think we have to go with an integrated approach, not relying on one component, but really using integrated approach. Genetics is important, microbes are important, inoculants are important. So I really go in an integrated approach rather than separate the approaches.
Yes. We have a long comment by Fatso Chongo and followed by a question. He's saying that mycorrhiza being obligate soil microbes and rely on the carbon from plants manufactured through photosynthesis. Can we say in this way, plants also help to combat climate change by trapping carbon dioxide?
Wow, interesting questions. Yes, that has been already demonstrated. I mean, yes, particularly in forest ecosystems, where mycorrhiza fungi can have up to 40% of what plant produces as carbohydrates, as photosynthesis. Yes, by boosting photosynthesis, yes, we can fix more carbon in global way. Yes, that has been documented, and mycorrhiza can play really a significant role in sequestrating carbon in the soil.
Yes, we have now a technical question, Mohammed, from Miriam Belfki. How you did separate between soil and rhizosphere microbiota in your test protocol process?
Well, this is typically really routine things. So, as I said, we define, for us, we took roots, then we separate the rhizosphere. Typically, we are just really shaking the root system to remove all the excess of soil. Then, in a bag, we are trying to mix in the soil to remove the rhizosphere. The root system is therefore washed 3 times, and at the end we use distilled water to remove excess of soil particles that are adhering in the root surface. But of course, there are all the time some microbes that are sticks really to the rhizosphere. We can't really avoid that we get rid of all the rhizosphere microbes.
Thank you, Mohammed. I invite the participants who were asking question at the beginning of the talk to please send it back, because people are sending question now, and I cannot see no more your question from the beginning. Please send them back. So we have a question from Sudarshan Yuta. Is there any proven qualitative improvement of produce along with yield improvement with use of microbes?
Yes, the answer yes. Antioxidant, protein contents, and and also some for medicinal active ingredient. Yes, I think that the use of microbes can help to increase quantitatively, or I would say the nutrition quality of fruits or vegetables.
Okay, thank you, Si Mohammed. We have a question from Abdelkader Rajai. I'm sorry for misspelling your name. Is it possible to use it for macronutrient biofortifications, and how? To use, of course, microbiome or microbiota.
So if I understand the question, the combination of micronutrient and bioinoculant for biofortification. If I understand it correctly, I think yes, we can. And in this industries, they are using some amendments other than microbial active ingredients.
Okay.
And how? There are different ways. So either seed coating or application in furrow or leaf applications. So there is many different applications, or even repeated applications during the growing season.
Okay. From Layla Taji, are there any studies that follow the impact of pesticides on the microbiome diversity and also on the behavior of microbes with fungi, especially as the soils are rich in pesticide residues?
The answer is yes. There are many studies with different, really targeting different group of microorganisms that documented the impact of pesticides on the microbial diversity. But also not only the pesticide, but sometimes this pesticide get degraded by microbes, and by degrading they release much more toxic compounds. Yes, you can find in literature studies that they have documented that.
Yes, a question from Jamal Urag is about the practices, the real practices in the agriculture in Africa, in Morocco. And he said, what about using microbial inoculants in Morocco and Africa? How to promote these practices, knowing the legal aspect linked to the introduction of microbe in nature?
Well, there are, commercial products I guess in Morocco and in Africa that they use bioinoculants. But as I said, as I showed here, we can also use different strategies using biostimulant or substances to somehow manipulate or increase microbial biodiversity. But I believe that both strategies are important for agriculture, and we should take them into account.
Okay, so a question from one of our colleagues, Rashid Al-Fatmi. He's at university, the same university as us. Do you have an example of specifically transferred gene RNA that correlated positively or negatively with the plant health improvements?
Well, not really, yes. We have many examples, and I have published on that, in which we have demonstrated that RNA system that has been transferred from cyanobacteria to arbusculum fungi, and we have also some examples that have been done between microbes and plants, positively or negatively. As far as I know, the studies didn't push that further, but there are studies documenting horizontal gene transfer between microbes and organisms. Although these events are rare, but for instance, in microbes, yes. The resistance to antibiotics. This is something really very common. Microbes exchange plasmids containing genes for antibiotic resistance. So then we can put it in a context. So it's a bad for health situation, but it's good in so it really depends on the context.
Thank you, Mohammed. A question from very technical. This one is from Mohamed Boulif, Professor Mohamed Boulif. Is this microbiota on seed in the soil or bare seed grown on petri dishes? Probably this question has not been completed.
Seed is in soil or bare. I don't. So I have really. I recommend you, Mohammed, to read. I think they have isolated some from but I can't. I'm sorry. I can't really. I don't know the details.
Okay. So I received a question from email from. Now I'm putting it. The question. Sorry. Yes, here's the question. This question is from Professor Ismail Kakmak. He sent by email just right now. Seaweed extracts are usually effective on plant growth in pot experiment under controlled conditions, but their effects are absent or minimum in field. How do you explain this?
Well, I a bit disagree. So for the examples that I know, if I'm giving the example that I know best, in Canada, so in wineries or in blueberries or strawberries, so these are really are getting really benefits in field conditions. So it's depend on the application. Number of application, is it leaf application or soil application? But as far as I know, the examples that I know from agribusiness plants, they are really getting benefits and applied in field conditions, not in the annual crops, but in perennial crops.
Okay. We have a question from Ado Yusuf. What is the application of combining bioinoculants, bacteria or mycorrhiza, with seed treatment chemicals? Do you see any negative effects, in other words, of the chemicals on the microbiome?
Yes, this technology exists already. So companies actually in many different countries are offering this. Because of course, if you are developing bioinoculants, you have to adapt yourself to agronomic practices of farmers. And typically, these bioinoculants are tested for compatibility with chemicals. So the technology exists, and it's already utilized.
Okay. So a question again. I think it's on the. Sorry. I'm trying to get you a new question, but it doesn't come back out. Let me show. Yes, this is a question again in the same subject. Chemicals, and so are there any studies that impact of pesticides on the microbiome diversity and also on the behavior of microbes with fungi? I think we have already answered this one, right?
Yes.
Oh, yeah, I have a lot. This is why. Yes, I'll give you this one from Ibrahim Ali. What are the implications of the increasing interest in microbiome research in the recent years for improved agriculture productivity? Probably Ibrahim Ali, Professor HIJRI answered partly this question in his presentation, but he will probably complete it now.
I mean, but typically it's really the last slide for conclusions. But of course, there are a lot of interest actually in all these technologies, and we believe that they can really get benefits and they can improve our production systems.
Okay, we have a question from Iyad Alawi. But I think it's from Latifa, basically.
Yes.
use the same study in the same field in 2 years, for example, do we expect the same results? She's talking about reproducibility of the results.
Well, the answer is yes. I would expect the same results, but we have to test it.
Okay. From Layla Taji, in your studies, did you take into account the presence of pesticide residues in the soil, or did you work with virgin soils?
Virgin soils. So soils was collected from a farm under organic farming since a long time ago. So it's an experimental farm of the province of the government that there is no pesticide that has been used for decades.
From Jean-Mélène Amari, can you give us some practices that we can apply in the smallholders farmers context where soil analysis is very rare? So how to manage the cropland microbiota without precise information about that? Very interesting question.
Interesting questions. I mean, smallholders farmers typically they are not using a lot of inputs. That is quite good already, and I would expect in that agro-ecosystems do much better in terms of microbial biodiversity than conventional agriculture system. So how to improve that? Probably improving soil fertility by organic amendments that can help to maintain or to increase cropland diversity.
Okay, thank you. I just want to highlight one thing before I get you to the next question, Mohamed, just for the people who are watching. So if you haven't the chance to start this webinar from the beginning, okay, don't worry, because this webinar will be broadcasted at the ESAFE UM6P channel. It will be broadcasted from the beginning. You can watch it from the beginning to find out what has been said by Professor Mohamed HIJRI, and you can watch it again and again, no problem. So, okay, so Mohamed Boulif, Professor Mohamed is back with a new question. Can you give us example of biological control of plant diseases, other microbes affecting plants?
Well, there are a lot of examples, a lot of examples. And the best example I can give you ever, it's for human health, nothing to do with agroecosystems. So C. difficile. So C. difficile, actually, it's really an opportunity. It's it's a pathogen that can compete very badly compared to indigenous community. So, and that's C. difficile. It's a real attack for people who were under antibiotic treatments. So we can do an analogy in agriculture. So the thing is, it's very difficult in terms of legislation because biocontrol agents will follow same regulations as pesticides. And the example I can give you is trichoderma harzianum, T22. That has been applied and is applied since decades for soil-borne pathogens, Botrytis, Fusarium, Verticillium, and other, and Sclerotinia as well.
Thank you, Mohamed. So a question from Khalid Arsey. After saying hello, is that postulate of the use of microbiota or biostimulants in general for all the soils and the geographically zones? So he wants to know if we can generalize this to all the soils, or it's dependent on the localization of the soil. I am talking about water and salt deficiency zones.
About water and salt deficiency zones. I don't really understand the question. Is that a postulate that you use microbiota or in general?
It's probably
I think yes. It really depends on the application. If we want to use some microbiota to alleviate the stresses of water deficiency and salt. Yes, I said mycorrhiza. Mycorrhiza inoculant has been proven its efficiency in these conditions. The answer is yes.
Okay, so we have a question from another colleague, Ashraf Al-Allali.
I know that.
You know him, so you have fans, Mohammed. So Ashraf is asking, where do you see the bottleneck that is holding large-scale research in the microbiota and soil metagenomes, and ultimately the use of biostimulants by farmers?
Well, it's a very good question. And I mean, it's very, very, very difficult to convince, farmers to use this. So farmers, you can see them as companies, and it's very difficult to change their agricultural practices. So this is a tough question. It's a real challenge. As I said, you have really to adapt yourself with them and probably go in really step by step. And you have to do some education. So the bottleneck is really in the field, and you have to do some education, and you have really to convince them and go in really gradually. But this is really a real challenge, no doubt about it.
Yes, Mohammed, I think this question is related to many other questions, and I cannot give them all to you, but I will summarize. It's about the difficulty of using this microbial inoculants for a simple small farm holder of farms. So is there any way that's really simply speaking, that someone is preparing a cocktail of microbiome by like in organic agriculture and using it to inoculate his plants? Is there any way, not really high technical, high scientific manner, that farmers can use this technology?
So these technologies, I think, yes, I get now the answer. Sorry, Ashraf, so I gave the wrong answer or a part of the answer. So as you see, it's very difficult even for farmers to do physical chemical properties of soil because of its cost. And these tools are still very costly, today speaking. I think, and also with scale. We're now in a field that's very heterogeneous, and microbial scale and agriculture scale are really very, very, very difficult. If I understood correctly the answer, so these technologies we are still really going further. We are not really to give or to offer a service to use these technologies to convince farmers to use biostimulants.
Okay, thank you. So we have Jihan Lanaya. She has two questions because she did first, as known, plant extracts are also biostimulants. Do you have an idea on the effect of plant extract on crop root microbiota? I think you have already.
Well, I think yes, it really depends on which extract, how it's done. So this is sound as compost or tea compost. So it's really depends. Yes, but it's potentially can act as biostimulant by bringing carbon to the soil, but also potentially some phytohormone. The answer is yes, but we have to test it, and it depends on which extract is available.
Okay, here is the second part of her question. Do microorganisms have an effect on the development of bulbs or corms?
The answer is yes.
Okay, so a question from Tabrika Elias. Can compost be considered beneficial to the soil microbiota? There is any study in Morocco? Is asking if there is any study about the effect of compost in soil microbiota.
The answer is yes, but in addition, compost bring another level of microbes, because compost as it owns contain already its microbial community that you are already adding. Yes, it increases microbiota richness in soil, and also it brings However, we have to be careful because also we can bring pathogens.
Okay.
So is there any studies of this in Morocco? I don't.
Sorry, I took off the question.
I think the second question was, is there any studies in Morocco for that? I think the answer is yes. There are a lot using compost, but as far as I know, no, not for microbiota business.
Okay. So a question from Vijay Kumar Gupta. It's from also the same university, Mohammed VI University Polytechnic. Is there any link between the dicer-like proteins in mycorrhiza and their role in the regulation of plant defense?
Well, good question. I assume there is one, but we have already to demonstrate this. So the dicer-like proteins in mycorrhiza are typically it's to protect mycorrhiza against virus attack. So probably the fact that mycorrhiza are living intimately with plants, probably it could be linked to plant defense. So I think that there is a link, but we have to demonstrate.
Yes, so it looks like Vijay is proposing a new research topic here.
So it's interesting.
Yeah, it is. Anthony Oyugbe is asking what quantity amount of the seaweed extract was applied in terms of kilogram per hectare or gram per pot?
Well, good question. So it really depends on the application on the crops. So seaweed, you can find it in liquid and in solid forms. And in liquid, you have different concentrations. So it's really depends depends on the products you are using, and also typically it's repeated applications every 3 weeks. So for my experiments, so I used very little amount. It's 3.5 milliliter per square meter. That is repeated for every 2 weeks. 3.5 milliliter is not that much, but I used a very concentrated product.
Okay, again, I just want to say again, if you have missed the beginning of the webinar of Professor Mohamed HIJRI, you can still retrieve it and watch it through the ESAFE channel. You have just to subscribe to the channel and you can go ahead and watch all the different webinars, mainly the one from Professor Mohamed HIJRI from the beginning. So a question from Samuel Ajeli. Microbial inoculants, biofertilizer, AMF, enhanced yields, and stress tolerance. Need for adoption and application in tropical agriculture.
Yes, I agree with you, Samuel. And we need to be patient. Patient because, I mean, time will come in which it will be democratized. So I think many colleagues, many industries are working on that perspective, and I think it's just a matter of time.
Okay.
But it will come.
Okay. So here is a student that I know very well. He's a PhD student, Ismail Mehdi. I like the students anyway. In terms of feasibility, he's asking, could biofertilization meet the world's need for food more than chemical fertilization?
Well, interesting question. So the answer, no, we are not. We still need chemical fertilizers because that counts for 50% for at least. I think, as I said, as an answer in other questions, we have to integrate both. Biofertilization is important, but chemical fertilization is important. So because we have to not only increase the production agroecosystem, but also we have to maintain, or to secure global food for the entire world. So my answer will be integrating both.
A question from one of the colleagues of Ismail, just Nidal Fahsi, who is the same lab and who is doing really good PhD project. He's asking, what are the most feasible and promising microbes for biofertilization, bacteria or fungi? In terms of your
very difficult to answer or to give a clear answer. It depends. So bacteria or fungi. So I would say yes, bacteria probably for some reasons. So it depends really on many parameters, formulations, applications, regulations. For fungi, I would say fungi are typically very easy to formulate the fact that they produce co-media. And I think here again, I am in favor of combinations.
Okay. Very specific questions about the policies and the regulations, Mohammed, from Jamal Urag. Given the absence of legal texts in Morocco, one should be very careful not to introduce very competitive microorganisms, which could irreparably change microbial diversity soils. What do you think?
Well, very good questions. Actually, I was planned to give a project or to show you a project about that. So definitively, when we introduce microbial inoculants, they may have an impact on microbial communities. However, this is a natural ecosystem. For agricultural ecosystem, anyway, it's unartificial, it's man-made. Agriculture field is not something natural. It's really highly anthropized, and we have actually, I have demonstrated for microbial inoculant. When we introduced them, we didn't observe any significant differences on the microbial communities in crop roots. But I think, yes, you are right, we have to be careful. Although in terms of microbes, so if we are speaking about some taxa, for example, Bacillus or Bacillus, there you can find them almost everywhere because we have this or this just stream. We have, winds that they can be as a vector to transport microbes from place to another place. But yes, we have to be careful, and it depends if it's a natural ecosystem. It's highly not recommended, but for agricultural ecosystem, this is a really an artificial system.
Okay, so I must say sorry for the people who are asking some questions that I'm not to Professor Hijri. It's just simply because that this question has been already asked. I'm sorry. So again, you can, if you would like to ask new questions, please go ahead. But we have a lot of them. We have Zineb Rishayed coming back, persisting in saying, how does the synchronization of the circadian rhythm, the light and the dark of mycorrhiza help the plants? Did you identify the molecular players involved in this synchronization and plant advantage of the temporal mycorrhiza?
Well, the question will, no, we don't have any evidence yet. But however, I will say the opposite. So the mycorrhizal fungi try somehow to synchronize their circadian rhythm with that of plants. So the plants has very sophisticated systems of photoreceptor that sense really the sunrise and sunset. And the fact that mycorrhizal fungi relies on carbon of plants, they have to synchronize their rhythm with that of plants. So I think it's a real determinant, but we don't have any evidence yet. Probably it might be that the synchronization can help the plants, but we don't have any evidence.
Okay. I find this question very interesting. This is why I'm giving it to you, Mohammed, from again Mohammed Boulif. How can we use crop rotation to modify soil microbiota, especially when it comes to fight soil-borne plant diseases?
I would say extremely important. Crop diversification, it's really crucial. As the system is diversified, as better is for microbial communities and for really reducing the impact of pathogens. As we have evidence actually in the literature, diversified rotation system with 3, 4, even 5 crops are much better than 2 or simplified rotation system. Definitively, I see a huge impact, important impact on crop diversification to combat somehow pathogens and to increase, soil microbial diversity.
Okay, so now I'm going to summarize a bunch of questions, and this is I'm pretty sure that that some of the students are waiting for such. So in terms of the research being undertaken on the microbiome and microbiota in plants, plant improvement and crop improvements, do you advise in terms of research where to dig? What are the questions that still to be answered, in the following years, that where the students can focus their research?
Well, tough question. I would say typically all the research really depends on fence. So this is really one of the important things. It depends on the fence, the term of the fence, short term, long term, and also the amount of fence. So these technologies are really very expensive. They require very skilled knowledge. They are requiring different expertise. I think I'm really going by recommend having integrated approach, working with agronomists, working with plant biologists, working with microbiologists, and yes, and going for really probably for long-term experiments. All the time, what I would also recommend, which is really important, we have first to identify what are the objectives and to formulate clear hypotheses. These are really the basis of any research before to be conducted in any different labs.
As an underlying question, how about educational programs in the subject, Mohammed? Are the academic community is building, developing such training program on this very interesting subject?
Well, Michel, very good question. I would say I was surprised when I saw that. I mean, we have published actually last year about this, and to summarize the story, I mean, fungi, they are relying. I mean, I'm talking about fungi, ascomycetes, basidiomycetes. I mean, light receptors, light sensing mechanisms are extremely important, and they are really the drivers that they are controlling their growth. So the best known system is neurospore across. It's a model that is well used. So either light, but also the temperature. So these are the two parameters that allows them to synchronize somehow to have their circadian rhythm. So coming from mycorrhiza, they're somehow inherited from their ancestor. So wild coral receptor. Wild coral receptor, it's sensitive to blue lights. And blue lights, it's typically in sunrise. And because the blue light doesn't have any penetration in the soil, so mycorrhiza fungi are able to grow somehow in the interface of the soil surface. In the 80s, a group in the United States, they have tested all the different wavelengths of light on the mycorrhiza growth for germination. And they have screened all the wavelengths, and they have observed that at a certain wavelength and a certain exposition, 10 minutes, typically between 10 minutes and 20 minutes, with blue light, fungal filaments are start branching. And that's a real story behind that. So one, Sunjay Lee, he's a former PhD student, started with this. That was really the beginning of the story. But of course, they have inherited that system from an ancestor.
Okay, we have a question from Basuri Suleiman. He's asking, can you give an example of a situation where microbe has improved water stress tolerance in plants? He is familiar with that of saline stress tolerance in quinoa. Any special mechanism involved?
Well, mycorrhizal fungi. So here in Morocco, many colleagues in Qadi Iyad, in other universities, deals with that when we have real evidences that mycorrhiza can really help to alleviate water stress. Unfortunately, for quinoa is one of the rare plants as brassica do that do not form mycorrhiza.
Okay.
So probably going with other options, trichoderma or other endophytes. So there are other participate endophytes probably could help.
So Amarjit Basra has a question about the inheritance. Is microbiome inheritable through seeds?
Yes, some of it. Yes, absolutely. Each seed has their own microbiota. Inherited, if I'm said as inherited, it really depends on that. So the question if it's really inherited from the mother, I will say no. I don't accept it. We have an evidence, but each seed has its own microbiota, and that microbiota, it depends on how seeds were treated. But inheritable, it's a good question though. So I didn't dig that deep.
Okay. Okay. We have a question from Shadi Berhar. What about the application of microbiota into in vitro cultures to produce healthy, certified plants to disseminate to farmers?
Yes, it's already done. Yes, I know. It's already done. Particularly, I will say in trees and nurseries, in vitro for fruit trees.
Okay, I think this question has been somehow answered, I guess. What from Therese and Europe? What are the most important genera of fungi and bacteria used in biofertilization?
Yes, I mean bacteria or rhizobia. Rhizobia, so it's a very heterogeneous group. It contains rhizobium, bradyrhizobium, and alpha proteobacteria. We have bacilli, so it's a group of bacillus. They are mostly used as biocontrol agents. We have azoospermillum as one of the most common one for free-living, nitrogen-fixing bacterium. For fungi, for example, for instance, we have mycorrhizal fungi, trichoderma, but we have some penicillium as well. So I mean, in terms of diversity, we have some good examples that are already way far away. Rhizobia still the most common bioinoculant commercialized so far.
Okay, we have a question from one of our young researchers from agrobioscience, Wissal Al-Haysoufi. She's a PhD student. She's asking, could we integrate bacteria and fungi too in such a biofertilizer? And she's asking if there will be any competition between them.
The answer is yes. And before integration, we have to test compatibility. If they are not compatible, forget about. But we have to do first compatibility tests, not only between bacteria and fungi, but also between bacteria. Bacterial strains must be somehow, should be compatible, not inhibiting each other. Same is true for between fungi. And it has to be done before coming with an integrated product.
Okay, so I would like to come back because one of the questions I tried to summarize from the other questions, but my microphone was muted. So we discussed, Mohammed, about the importance of microbiota and the research and the scientific, activity in microbiota. And I asked, and people were asking, how about educational programs in such an interesting topic? What are there any actually training program, master degree, bachelor degree in the world that treating such a topic?
I think so, and this is something in the ESAFE we are working on. So thank you, yes, and we have really to integrate. So as really a training program dedicated to that. As far as I know, I don't know about any program, but different institutions, of course, they have different group of research. There are really thousands over the world that they are working on these topics. I'm not alone. So there is really a myriad of research group that they are working on that topics. So for training format, I think yes, it might exist.
Okay. We have a question from Amogu. So he's asking about the large-scale application of this combination. Are there biostimulants formulated from plant extract and rhizobacteria used as a large-scale bioproducer?
I think so. And mostly those that they are commercialized, most of the time, the formulation is trade secret. You don't know what is in. But I think yes, they might be using some plant extract, for example, to control some pathogens, or some phytohormones, or some peptides, or whatever. But I will say yes. But then for the biostimulants, most of the time I would say it's a secret formula.
Okay, thank you, Mohamed. We are approaching now somehow the end of the webinar. This is why I will take 3-4 questions, no more. The first one is from Miriam Belfki. You do mention that microbiota phosphate has a positive effect on plant signals. Can you give us the major metabolic pathways related to plant growing, which are enhanced by the microbiota phosphate activity?
Phosphate has a positive effect on plant signal. Major metabolic pathways related to plant growth, which are enhanced by microbiota activity. So the question is not clear to me. The positive effect.
We can ask Miriam, the participant, to rephrase the question or clarify it. But in the meantime, I can give you another question if you like.
Which are enhanced by microbiota activity. Well, I think P is an essential element for plants, and not only for plants but for all living organisms, and they are really in all the major metabolic pathways of plants: energy, structure as a phospholipids, ATP, ADP, plant signaling pathways via phosphorylation, dephosphorylation of protein via kinases, and also for DNA, RNA. So I think yes, mycorrhizal fungi are very efficient in solubilizing and uptaking phosphate, providing it to the plant, and it enters the major, primary metabolic pathways of plant cells.
Okay, thank you, Mohamed. A question from Pierre Pascal Dien. Is there any issue? You have touched on the GMO, and you said that you are not doing any genetically modified organisms in your presentation. He's asking, is there any issue of using GMO on microbiota community, not on the plants?
Well, this is a very good question. I know the answer. I can spend hours giving that answer. GMO is only typically used for crops, for plants, but for other animals. And in microbes, as I said, have a very sense during their evolution are able to exchange their genes. The best example I can give you is plasmids. So plasmids are mobile elements. They can really jump from taxon to taxon. So GMO on microbes, or particularly what microbes speaking, I assume that it's for prokaryote. So it's yes and no. So we can have some improved systems by using some plasmids, some transformations. And that transformation should be considered as GMO? I don't think so. So it's really depend on the context, but engineering a new microbes or not is not a good way to go, but improving some traits using conventional genetics, yes.
Okay. I hope it's fair for the participant now. I think you know this student, Zakaria Al-Harari. What do you think, instead of adding microbes as a bioinoculants, we use substances which are part of the established communication to improve crop production? He's referring to
I think it's a good idea. I think it's a good idea. Why not?
Has this been done before, Mohammed?
I think so. I think so.
Okay. Okay. So let me give you the one, not the last one, but it's a question from Hassan Bouksim. He's concerned about the transfer to market, taking into consideration the coupling between biology and soil physics. How do you see this transfer of this technology to the market, where you need always to couple the what is in terms of biological function of the soil and its structural physical structure?
I think it's a good question, and it's still really challenging. So we have, as I said, we have to do some education, but also good science. I think if we are doing good science and we are generating extensive knowledge, I think it's easy to convince, the end users to integrate that technologies. But definitely, soil physics, physical chemicals, not only physics, but also is important. And I believe, and it's already done in some countries, we have really to adapt our product depending on type of soils and climate conditions.
Okay.
But I think some countries, they are doing really quite well, that they are developing already biologics, that they are specific for sandy soil versus some product that they are efficient for rich organic matter soils.
Okay, I think we're getting to the last question. Is from Christian Adler Fars. I think you have already touched on this one. Good. He thanks you for the good presentation. And his question is, did you use the individual active ingredient or the crude extract for what you have presented as a result?
Yes, thank you. So what we have used is really the crude extract. So it's a commercial product. So the company, they are really using ascophyllum as a raw material that is collected from the coast, and then I think they process it. They have, their own process, and all this non-soluble organic matter is formulated as a seaweed extract. But I think they exploit some nutraceutic compound that they are extracted, and the remaining non-soluble part is used to formulate that product.
Okay, thank you, Mohammed. Just as a concluding remarks, so I would like you to just elaborate on what are today the barriers that are today for this the result of this research to come to the real world. So I know that you have already touched on a little bit in terms of practices and in terms of technology. So are these still big barriers, or are we really close to the markets with these new technologies, and the smallholder will benefit from it?
Well, I'm optimistic. So I think we have all the technologies and all the knowledge and even momentum. Actually, with this COVID-19, we need to think about our agro-ecosystem as a whole. And I think we have also to bridge or to make a way to improve connections between academia, research institutions, industries, and field applications. So if we have to do some work, we have really to make or to bridge these components.
Okay, thank you, Mohammed. Thank you again, Mohammed, for this really interesting topic that you have nicely explained and tried also to simplify as much as you can. Thank you for the efforts that you made to get this as close as possible to many of the people who are participating. I was really very happy with the discussions, also with the questions you have provided to the people. A lot of people are still asking questions. I'm sorry, we cannot take all of them. Again, you can still watch the webinar by Professor Mohamed HIJRI at the UM6P ESAFE channel at YouTube anytime. Again, you can still ask questions. We will address them to Mohammed after the webinar. Thank you again, Mohammed, for accepting this invitation. And just give you some information now for the next speaker of this webinar series. My next, sorry, I have to show. Yes, my next speaker, I will invite Professor. Sorry, no. Yes, Professor Kuku Amuzu. He will come for a webinar on next Monday, next week, 2 p.m. And his webinar will be on elaborating on plant nutrition strategies for climate change adaptation in Africa. So just very quickly, Professor Kuku Amuzu is from the African Plant Nutrition Institute, which headquartered in Morocco, close to the University Mohammed VI Polytechnic. So please be our guest for next Monday to watch and to participate in this webinar. A lot of people were asking us to somehow, sorry. Somehow provide them certificate for participation. So this is very difficult for us because there is a lot of participants. It's a big logistics to produce, but we will think about it. We'll promise. So last and not least, I just want you to know that ESAFE, the School of Agriculture of the University Mohammed VI Polytechnic, is organizing and developing and recruiting for a new cohort for a master's degree in fertilizer science and technology. So if you would like, as a student, you have a bachelor or you have an engineering degree and you would like to apply for this master's degree, please go ahead. You can do it by sending a request at the email highlighted here, esaf@um6p.ma, or through WhatsApp discussion with this number in Morocco. Okay, you will have one of our master degree coordinator, Mr. Ismail Ghorban, will be answering your question and will be taking all the requests from you. Again, thank you very much for the for your participation. Don't miss next Monday the webinar by Professor Kuku Amuzu from the African Plant Nutrition Institute, and his seminar will be on plant nutrition strategy for climate change and adaptation. But before closing this webinar, I would like again to thank you for the participation. Thanks to all the colleagues who are behind all this, that are in the back office, who are organizing all these webinars. Thanks to Mr. Osama Al-Qasimi from our group, from our team, who is organizing and making sure that everything is working behind. Thanks, Osama, for the great job you have done since the beginning of this webinar. Thanks for our thanks again for our guest today, Professor Mohamed HIJRI, and thanks also for our strategic and partners, OCP-Africa and RUFORUM. With that, again, thank you, and I see you for the next webinar on Monday next week. Bye-bye.
Bye-bye. Have a good day.
You too, Mohammed. Bye-bye.